This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas.
Okay, so get this. We are diving into some seriously sci-fi stuff today. Scientists, get this, they created a mouse with two fathers.
Yeah.
Like two biological dads.
It's really quite remarkable, isn't it? They managed to overcome what was considered like a fundamental law of nature, with mammals.
Yeah.
That you need both a mother and a father to create offspring.
Yeah, so we've got this press release detailing how they pulled it off.
Huh.
And it's packed, it's dense, a lot of scientific info in here.
Right.
So our mission today is to break it all down, for you listening, unpack how they did it, what it could mean for the future, like for stem cell research, regenerative medicine.
Mm-hmm.
And of course, we can't ignore the massive ethical questions this all raises.
Oh, absolutely. This isn't just about creating mice with two dads. It's about, potentially rewriting the rules of reproduction as we know it.
Absolutely, yeah. So this actually wasn't the first attempt at creating a bi-paternal mouse.
Okay.
Scientists have been trying to crack this nut for a while. But previous attempts always seemed to hit a wall, like the embryos would just stop developing at a certain stage.
Right.
What was the key to finally getting past that?
I think the key was cracking the code of something called imprinting genes.
Okay.
You can think of them like genetic switches that control how other genes are expressed.
Okay.
And for a long time, these imprinting genes were considered, the main reason why mammals couldn't reproduce with two parents of the same sex.
Okay, so break that down for me a little bit more. Like, how do these imprinting genes work?
So essentially, they act like a set of instructions that determine which genes from each parent are activated or silenced in the offspring.
Okay.
It's a very delicate balance, and if it's disrupted, then development goes haywire.
So it's not just about having the right genes, it's about having them turned on or off at the right time or
In the right combination.
in the right combination, yeah.
Precisely, yeah. And the researchers actually quote one of their colleagues in the paper, Ki Zhu, and he sums it up perfectly. He says, the unique characteristics of imprinting genes have led scientists to believe that they are a fundamental barrier to unisexual reproduction in mammals.
Mm-hmm.
They were basically saying this is why same-sex reproduction doesn't happen naturally in mammals.
Okay, so they identified the roadblock, but how on earth did they actually overcome it?
Right.
This sounds incredibly complicated.
It was they targeted 20 specific imprinting genes.
Wow.
And they used some seriously high-tech gene editing techniques to modify them.
Okay.
I mean, we're talking about tweaking the very blueprint of life here.
Hold on. So if scientists could already clone mice, why is this breakthrough with imprinting genes such a big deal?
Yeah, it's a great question.
Mm-hmm.
Cloning is about creating a genetic copy of an existing individual.
Right.
This research is about creating life from two sets of, paternal DNA.
Right.
Essentially rewriting the rules of how that DNA is used to build an organism.
Oh, okay. So it's like they hacked the system and bypassed the usual requirements for creating a mammal embryo.
Exactly. And that's what makes this research so groundbreaking.
Yeah.
By manipulating these imprinting genes, they were able to create viable embryos from two fathers.
Yeah.
It opens up a whole new world of possibilities.
Okay, so let's get into the nitty-gritty. What did they actually do to these genes? Did they swap them out, delete them, give you the details?
They used a whole toolbox of gene editing tricks.
Okay.
Things like frame shift mutations, which disrupt the gene's code.
Okay.
Gene deletions to remove them entirely.
Okay.
And even edits to the regulatory regions that control how the genes are expressed.
Wow.
It was a very delicate and complex process.
Yeah, it sounds like they really had to fine-tune the system.
Yeah.
Like, I can only imagine how many attempts it took to get it right.
You're right. It wasn't easy.
Yeah.
They mentioned in the press release they had to experiment with different combinations of modifications to
Right.
find the ones that actually worked.
Wow, so they basically rewrote the rules of reproduction at the genetic level.
Mm-hmm.
It's mind-blowing, but I'm guessing it wasn't all smooth sailing.
No, you're right. It wasn't a perfect process.
Yeah.
They had a pretty low success rate.
Huh?
Only about 12% of the viable embryos actually made it to birth.
Wow.
And even then, not all of them survived to adulthood.
So even with all this advanced gene editing, they still face some serious challenges.
Absolutely. Mammalian development is incredibly complex.
Yeah.
Even tiny changes to the genetic code can have ripple effects.
Right.
They found that some of the pups had developmental defects and shorter lifespans, likely because of the modifications they made to the imprinting genes.
Okay, so it wasn't a walk in the park.
Right.
What about those pups that did make it to adulthood? Were they able to, reproduce normally?
That's where things get even more interesting.
Okay.
The mice that lived to adulthood were all sterile.
Oh, wow.
They couldn't have offspring the natural way.
So even though they were born from two fathers, they couldn't pass on their genes to another generation.
That's right.
Okay.
But here's the twist.
Okay.
Even though they were sterile, the study found that these bi-paternal mice actually had increased cloning efficiency.
Hold up. They couldn't reproduce naturally, but they were better at cloning. I'm not sure I follow.
Yeah, it seems counterintuitive, right?
Yeah.
But it highlights just how complex the relationship is between genes, development, and reproductive potential.
Yeah.
The modifications to the imprinting genes, while hindering normal reproduction, might have inadvertently created a cellular environment that was more conducive to cloning.
So it's almost like they traded one reproductive ability for another.
Mm-hmm.
Fascinating, but also kind of a head-scratcher.
It definitely raises more questions than it answers, but that's what makes this research so exciting,
Yeah.
It's pushing the boundaries of our understanding and opening up new avenues for exploration.
Right, so we've talked about the science, the challenges, the surprising twists.
Right.
But now let's zoom out a bit and talk about why this research matters.
Okay.
This breakthrough has huge potential for stem cell research.
Yeah.
Remember those imprinting genes we talked about? Well, it turns out those same genes play a crucial role in determining how stem cells develop and what types of cells they can become.
Exactly.
Okay, remind me, why are stem cells so important? What's all the hype about?
So stem cells are like the blank slates of the body.
Okay.
They have the potential to develop into any type of cell, heart cells, brain cells, you name it.
Oh, okay.
And that makes them incredibly valuable for things like regenerative medicine,
Right.
where we're trying to repair or replace damaged tissues and organs.
So by manipulating these imprinting genes, are you saying scientists might be able to create even better stem cells?
Exactly. The modifications to those imprinting genes led to stem cells with something called more stable pluripotency.
Okay, you're going to have to break that down for me. What exactly does more stable pluripotency mean, and why is it so important?
So pluripotency is the ability of a stem cell to develop into any type of cell in the body.
Oh.
It's what makes them so versatile.
Gotcha. So the more pluripotent a stem cell is, the more potential it has to become any cell you need.
Exactly.
Gotcha.
But sometimes stem cells in the lab, they lose their pluripotency over time,
Sure.
or they might not be as versatile as we need them to be.
Okay.
This research suggests that tweaking those imprinting genes could be the key to creating stem cells that are more stable, more reliable, and more effective for therapeutic applications.
This is starting to sound like the stuff of medical miracles.
Yeah.
Are we talking about growing new organs in the lab, repairing spinal cord injuries, that kind of thing?
Potentially, yeah. Imagine being able to take a patient's own cells, create stem cells from them, and then use those stem cells to repair damaged heart tissue after a heart attack, or to grow new brain cells to treat Parkinson's disease.
Wow.
That's the kind of future this research points towards.
That's incredible. That sounds like we're on the verge of a revolution in medicine.
It's certainly a significant step forward.
Yeah.
And the lead researcher, Wei Li, actually says in the press release, this work will help to address a number of limitations in stem cell and regenerative medicine research.
Wow.
They're clearly very optimistic about the potential.
Okay, so we've got this incredible breakthrough with huge potential benefits for medicine.
Right.
But I know there's another side to this coin that we can't ignore.
Yeah, you're absolutely right. This research raises some significant ethical questions that we need to grapple with.
Let's dive into those ethical considerations next time.
Okay.
We'll be right back with more of this deep dive into the world of bi-paternal mice, stem cell research, and the ethics of it all.
Thank you for being curious and subscribing, following, liking, rating, and reviewing our podcast episodes. Your support really helps build a vibrant Heliox community.
All right, so we're back, and I think it's time to tackle the elephant in the room.
Okay.
The ethics of all this. I mean, creating a mouse with two fathers is one thing, but what about the potential for applying this technology to humans?
Yeah.
It feels like we're stepping into some seriously uncharted territory.
Yeah, you're absolutely right.
Yeah.
This research raises a whole host of ethical considerations that we, as a society, need to carefully consider.
Mm-hmm.
It's not just a scientific question.
Right.
It's a philosophical and societal one.
Absolutely. So before we get too deep into hypotheticals,
Right.
let's ground ourselves in the reality of where things stand right now.
Okay.
This research is still in its very early stages, right?
Exactly. We're talking about a single study in mice.
Right.
And even then, the success rate was pretty low.
Yeah.
And the mice that did survive, they often had health issues.
Right.
It's a long way from being a safe or reliable technology, even for mice,
Right.
let alone humans.
So we're not exactly on the cusp of designer babies with 2 dads or 2 moms.
No, not by a long shot.
Okay.
There are still huge scientific hurdles to overcome before we could even consider applying this to humans.
Right.
And even if those hurdles were cleared, the ethical implications would be enormous.
Okay. So let's say, hypothetically, that this technology does advance to the point where it could be used in humans.
Okay.
What are some of the ethical questions we need to be thinking about?
Well, for starters, there's the question of consent.
Right.
Would a child conceived using this technology be able to consent to being born with this kind of genetic manipulation?
Right.
And what about the potential psychological impacts on individuals born through this method?
Yeah.
These are uncharted waters, and we need to proceed with extreme caution.
It also makes me think about the potential for this technology to exacerbate existing inequalities.
Yeah.
Would access to this kind of reproductive technology be evenly distributed? Or would it further widen the gap between, the haves and have-nots?
That's a crucial point.
Mm-hmm.
We've already seen how new technologies can sometimes deepen existing inequalities.
Yeah.
It would be crucial to ensure that any advancements in reproductive technology are accessible to all,
Right.
regardless of socioeconomic status.
Absolutely. And then there are also the broader societal implications to consider. How would this technology impact our understanding of family, parenthood, and what it means to be human?
Yeah.
These are profound questions that go beyond the realm of science and touch on our deepest values.
Exactly. This research forces us to confront some, fundamental questions about what it means to be human and how we define family.
Mm-hmm.
It challenges traditional notions of kinship and forces us to rethink our understanding of reproductive rights and responsibilities.
It's a lot to unpack, and it's not like there are easy answers to these questions.
You're absolutely right. There are no easy answers.
Right.
But that's precisely why it's so important to have these conversations now.
Right.
While the technology is still in its infancy,
Yeah.
we need to engage in thoughtful and informed public discourse to ensure that any future developments in this field are guided by ethical considerations and societal values.
It's a reminder that science doesn't exist in a vacuum. It's always intertwined with ethical, social, and philosophical questions.
Precisely. And as scientists continue to push the boundaries of what's possible, it's crucial that we as a society keep pace with those advancements and engage in thoughtful dialogue about their implications.
Absolutely. And speaking of pushing boundaries, one of the details in the press release that really caught my eye was the fact that these bi-paternal mice, despite being sterile, were actually better at cloning.
Yeah.
Can you shed some light on that?
Yeah.
Seems like a bit of a paradox.
It is fascinating, isn't it? It suggests that while the modifications to the imprinting genes hindered normal reproduction, they might have inadvertently enhanced the process of cloning, which, involves transferring a nucleus from one cell to another.
So it's possible that by tweaking these imprinting genes, they actually created a cellular environment that was more receptive to cloning.
That's one possibility.
Yeah.
It could be that certain imprinting patterns are more conducive to cloning than to natural reproduction.
Right.
It's a reminder that we're still just beginning to understand the complex interplay between genes, development, and reproductive potential.
It seems like with every answer we uncover, 10 new questions pop up.
Absolutely.
It's a reminder of just how much we still don't know.
Absolutely. And that's the beauty of scientific exploration.
Yeah.
It's a never-ending journey of discovery, constantly pushing the boundaries of our knowledge and understanding.
So we've explored the science, delved into the potential applications, and grappled with the ethical considerations.
Right.
But I think it's important to remember that this research isn't happening in isolation.
Yeah.
It's part of a much larger scientific landscape that's constantly evolving.
You're right. This research is just one piece of a complex puzzle.
Hmm.
And to fully appreciate its significance, we need to consider it within the context of other advancements in fields like genetics, stem cell research, and reproductive technology.
It's like we're standing on the edge of a new era of scientific discovery,
Yeah.
with the potential to transform our understanding of life itself. But with that potential comes a profound responsibility to ensure that these discoveries are used wisely and ethically.
I couldn't agree more.
Yeah.
We have a collective responsibility to engage in, open and honest conversations about the implications of these advancements.
Yeah.
It's not just up to scientists,
Right.
it's up to all of us to shape the future of these technologies.
I think this deep dive has been a great example of how to approach complex scientific topics with both curiosity and critical thinking.
Yeah.
And we've explored the excitement of the breakthrough, but we've also been mindful of the potential risks and ethical challenges.
And hopefully our listener has come away with a deeper understanding of the science, a sense of wonder at the possibilities,
Yeah.
and a renewed appreciation for the importance of engaging in thoughtful discussions about the future of science and technology.
Well said, but before we wrap things up,
Okay.
I have one last question for you.
Right.
We've talked a lot about the potential for this research to advance stem cell therapies and regenerative medicine.
Right.
But what about the bigger picture? What other seemingly impossible biological barriers might we break down in the future?
That's a question that should spark the imagination of anyone who's listening.
Yeah.
Because the truth is, we're living in an era of unprecedented scientific discovery.
Right.
What seems impossible today might be commonplace tomorrow.
You know, it's amazing to think about how much our understanding of biology has changed, even in the last few decades.
Yeah.
Like, what seemed like science fiction is now becoming reality.
Hmm.
And this breakthrough with bi-paternal mice is just one example of that.
It really highlights, how quickly the field of genetics is advancing.
Yeah.
We're gaining the ability to manipulate the very building blocks of life in ways that were unimaginable just a few years ago.
And as we unlock these incredible capabilities, it's more important than ever to be having these conversations about the ethical implications.
Oh, absolutely.
You know, it's not just about what we can do, but what we should do.
It's crucial to have, a thoughtful and informed public dialogue about the potential consequences of these advancements. We need to consider not just the scientific aspects, but also the social, ethical, and philosophical implications.
This research with the bi-paternal mice really forces us to confront some fundamental questions about parenthood, identity, and what it means to be human. It challenges our traditional notions of family and reproduction.
It does, and it raises the question of where we draw the line.
Right.
If we can manipulate imprinting genes to create a mouse with two fathers,
Right.
what other boundaries might we be tempted to push?
It's a slippery slope argument, but it's a valid one.
Yeah.
Where do we set the limits on scientific exploration, especially when it comes to something as fundamental as human reproduction?
That's a question that society as a whole needs to grapple with.
Right.
It's not something that scientists can decide on their own. It requires a broad conversation that includes ethicists, policymakers, religious leaders, and the public at large.
It's a reminder that science doesn't exist in a vacuum.
Right.
It has the potential to impact every aspect of our lives, so we need to be thoughtful about how we guide its development.
Exactly. This research has, it's opened a Pandora's box of possibilities and challenges.
Yeah.
It's a powerful testament to human ingenuity.
Mm-hmm.
But it also serves as a stark reminder of the complex ethical dilemmas we face as we venture further into the uncharted territories of genetic manipulation.
Well, I think we've done a pretty thorough job of unpacking this groundbreaking research.
Yeah.
We've gone from the intricate details of imprinting genes to the mind-boggling potential of stem cell research and the profound ethical questions it raises.
And hopefully our listener has come away with a deeper understanding of this incredible scientific breakthrough.
Right.
And a sense of wonder at the possibilities that lie ahead.
Mm-hmm.
But it's also a call to action.
Yeah.
We need to stay informed, engage in these critical conversations, and help shape the future of science and technology in a responsible and ethical way.
I think that's a perfect note to end on.
Yeah.
This deep dive has been a real eye-opener, and I hope it sparked some curiosity and critical thinking in our listeners.
It's been a pleasure exploring this fascinating topic with you.
And to our listener, we encourage you to keep exploring, keep asking questions, and never stop learning. We'll be back soon with another deep dive into the most fascinating and thought-provoking topics of our time.
Sounds good.
Until then, stay curious. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.